Alright, so let's continue with glycolysis proper. So glycolysis, we already established that glycolysis simply means you want to split glucose into two equal half of pyruvate with the sole aim of getting ATP. That is the reason why you are doing it. That's the reason why we're doing glycolysis. And we established that it occurs in the cytosol or cytoplasm for all cells. Is that not so? Alright. Now today what we want to discuss proper is the pathway of glycolysis. Now I will just take a simple red blood cell here, and I will assume that this red blood cell wants to do glycolysis. Are you there with me? Now you will agree with me that glucose will need a transporter to enter into this red blood cell. Is that not so? And I believe you want to say GLUT1. Is that not so? Because I said GLUT1 is found in all tissues, and it is for the basal uptake of glucose. Are you there with me? Meaning that it has a low Km, which translates to high affinity. Is that not so? Let me not to bother you. But the transporter here in the red blood cell is GLUT1. Now glucose will enter the red blood cell. The thing is that if glucose enters this red blood cell, it has the propensity to leave the red blood cell again. So what we do is that we try to trap the glucose inside the red blood cell. How will you trap the glucose? By phosphorylating it. So my first step of glycolysis is that I have glucose being converted to glucose 6-phosphate. Is that not so? Now when I added the phosphate to glucose, it becomes charged, it becomes negatively charged, so it cannot leave the cell again. The question is, from where do I have this phosphate, or from where did this phosphate come from? And I will reply you by saying that it comes from here, from ATP. So ATP reacts, then we have ADP. This is step 1 of glycolysis. Are you there with me? So the first step of glycolysis is to phosphorylate glucose so that it's not going to leave the cell. Now which enzyme is responsible for this step? The enzyme is called hexokinase or glucokinase. Any of them, hexokinase or glucokinase. Now hexokinase will be in all the cells, majority, like in all the cells basically contains hexokinase, while specifically for the liver, that's where you will see glucokinase. So if we are to draw a table trying to differentiate between hexokinase and glucokinase, you will discover that while hexokinase is found in all cells, glucokinase is found only in the liver. Just to repeat, the first step of glycolysis is to trap glucose inside the cell. Is that not so? So we have glucose 6-phosphate. Which enzyme does that? Hexokinase or glucokinase. Hexokinase found in all cells, glucokinase only in the liver. As we proceed, I will explain the difference between hexokinase and glucokinase. Before we do that, acknowledge the fact that we utilize one molecule of ATP in step 1. Is this not contrary to the aim of glycolysis? The aim of glycolysis, we said, is to get ATP. Now in step 1, we already utilize one ATP, meaning we are going against our aim. Are you there with me? But just be calm. Let's proceed. This is for step 1. Mind you, I said I will come back to differences between hexokinase and glucokinase. I just gave you one, that while one is found in all cells, the other one is found majorly in the liver. Now what are we doing in glycolysis basically? We are splitting glucose, right? But what is the first step we did now? We just added phosphate to glucose. Now can we split the glucose now? Can we split it now? Let's see. Let me draw glucose 6-phosphate for you. Here's carbon 1, 2, 3, 4, 5, and carbon 6. Now if this carbon 6 now, there's already a phosphate here, right? PO4. There's already a phosphate group there. Now the thing is, if we want to split this glucose, this is the pyranose structure of glucose. If I want to split it, I will split it along this plane where the oxygen is. And if I should do that, it will not be symmetrical. What do I mean? On this side we have 2 carbon, on this other side we have 4 carbon and phosphate and 1 phosphate. So what should I do now? I will try to make it symmetrical so that once I split it, both sides will be equal. That's why the next step in glycolysis is to isomerize glucose 6-phosphate to fructose 6-phosphate. So what I get will be what? Fructose 6-phosphate. Are you there with me? So if I'm to draw fructose 6-phosphate for you now, you will discover that here will be the structure. It's called the Furano structure. Here I have carbon 1. Here I have carbon 6. So here is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. On this carbon 6 now there is the phosphate. Is that not so? That's why it's called fructose 6-phosphate. Is that not so? Alright. Now can I split it now? Can I split? Let me split along the plane of the oxygen. On this side I have 3 carbon, 1, 2, 3. On this other side I have 3 carbon also, 1, 2, 3. So 3 carbon each. But one thing you will notice is that on this other side there is what? There is 1 phosphate attached there. But this side does not have what? Does not have the phosphate. Are you there with me? So meaning we can still try to make it symmetrical before we split. Come back to what I'm trying to tell you. Initially we trapped the glucose in glucose 6-phosphate. Then we attempted to split it, but we discovered that it is not symmetrical. One side is having 4 carbon and a phosphate, the other side is having just 2 carbon. Then we try to make it more symmetrical by undergoing a reaction called isomerization. That enzyme is called isomerase enzyme. Shall we put it here? Isomerase. So you might want to say glucose 6-phosphate isomerase. I get my point. That will be the name of the enzyme, isomerase enzyme. Now, when we carry out that reaction, we discover that on this side we're having 3 carbon, on this side we're having 3 carbon and a phosphate, meaning it's still yet not symmetrical. Let's proceed. The next thing to do now is to make it what? Symmetrical by adding what? 1 more phosphate to this side here. By adding 1 more phosphate to this carbon here. Now when I add the phosphate to this carbon, what I will get will be, remember this is carbon 1, is that not so? Then we have what we call fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. Are you there with me? Fructose 1,6-bisphosphate. Okay? The name of the enzyme that catalyzes. Now, if you, once I add it now, you notice that it's now what? Symmetrical, meaning we can now split. But before we split, let's put in our enzyme. The enzyme for step 1 again is hexokinase or glucokinase. The enzyme for step 2 is isomerase enzyme. The enzyme for step 3 is what we call phosphofructokinase 1. Phosphofructokinase 1, in short, it is called PFK1. PFK1. You there with me? Alright, so the first thing, we added phosphate to glucose to make it glucose 6-phosphate, done by hexokinase or glucokinase. The next thing we realized that, oh, let's split our glucose 6-phosphate, but we discovered that it's not symmetrical, so we undergo a reaction called isomerization reaction, in which we now form fructose 6-phosphate. Again, fructose 6-phosphate is still not symmetrical, so we added 1 more phosphate to the carbon 1 and we got fructose 1,6-bisphosphate. Now, this phosphate we added to carbon 1, from where did it come from? It came from here, from ATP. So again, we have the reaction ATP forming ADP. Are you getting my point? ATP forming ADP. Are you getting it now? Now you notice that here is step 2, here is step 3. And you notice that for both step 1 and step 3, we utilize ATP. which is against glycolysis. We said our aim is to produce ATP. Are you there with me? Alright. Now, that's why the first 3 steps, this step 1, step 2, step 3 of glycolysis are termed as investment phase. They are termed as what? As investment phase. Let me erase this now so I write it for you. They are termed as what? As investment phase. They are termed as investment phase. Investment phase. So step 1, 2, 3, investment phase. Why is it called investment phase? Because in this phase, instead of us producing ATP, we end up using ATP. Are you there with me? Alright. Now, before we proceed, because the next phase we want to enter is called the splitting phase. I get my point. Before we proceed, there's something you need to know. This step 1 here is irreversible. Step 1 is irreversible. Step 3 is also irreversible. In short, in the entire glycolysis pathway, 3 steps are irreversible. 3 steps are irreversible, and we've seen 2 just now. Step 1 catalyzed by hexokinase or glucokinase. Step 3 catalyzed by PFK1. The last one we will see towards the end. Are you there with me? Alright. So before I proceed, I would like to summarize the points in this investment phase, just for a proper understanding. I said the investment phase is the phase whereby instead of us producing ATP, we end up what? Investing ATP. We end up using up ATP. And the step 1, just write with me, is catalyzed by what? Hexokinase or glucokinase. The step 2 is catalyzed by what? An isomerase enzyme. And the step 3 is catalyzed by what? By phosphofructokinase 1. I also make mention that the first thing to do to glucose once it enters the cell is to what? Is to phosphorylate it. That was what hexokinase or glucokinase does for us. So that also are the 6 carbon. Then we try to split. We notice that it's not symmetrical, so we use our isomerase enzyme to make it symmetrical. Again, we discover that it's still not yet symmetrical. We now use the next enzyme, which is phosphofructokinase 1. This enzyme, this first enzyme, is irreversible. This one is also irreversible. This one uses 1 ATP. This one also uses 1 ATP. Okay. Now, before we move to the next phase, which is the splitting phase, I would like to chip in something. There is a difference between hexokinase and glucokinase. So let's just do it now. Differences between hexokinase. So difference between hexokinase and glucokinase. Okay, so let's do it sharply. On this side, I will put hexokinase for HK and glucokinase for GK. I already give you one difference already. What is that difference? I said this one is found in all cells, right? This one is found majorly in the what? In the liver. Is that not so? Then this one has what we call, see, if we're talking about enzyme, the only way we describe enzyme is by their Km and Vmax. So let me just put Km here. Let me put Km here. Let me put Vmax here. Let me put what? Vmax here also. I get my point. Now, the way we describe enzymes by Km and Vmax. So, hexokinase has what we call low Km. Why this glucokinase has high Km? Again, hexokinase has what we call a low Vmax. Why glucokinase has a what? A high Vmax. Are you there with me? A high Vmax. Okay. Now, Km basically tells us about the affinity of the enzyme for substrate. Are you getting my point? The lower your Km, the more affinity you have. Meaning, in terms of affinity, I can say that hexokinase has a high affinity, while glucokinase has a low affinity. Okay, meaning glucokinase we need a high amount of substrate before it will start working for you. On the other hand, hexokinase with just low amount of substrate it can work for you. That's why we call it that's a low Km. And since it can work for you even when you give it just a low substrate, it means it actually loves the substrate more, meaning it has a high affinity. Are you there with me? How do you remember this? H for humble. H for humble. Are you there with me? Okay. Meaning if you are humble, it will be low, right? So low Km, low Vmax. The other would be for what? The opposite will be for what? Glucokinase. That is for that. Okay. So with this now, I'm done with the investment phase of glycolysis. You know these things very well. Later on, we will talk about the regulation of the pathway. But first of all, we need to finish everything. We need to finish everything first before we think of regulation. So the next phase I want to enter will be the phase called the splitting phase. The splitting phase. Okay. Now, where do we stop? We stop at fructose 1,6 bisphosphate. So we have fructose 1,6 bisphosphate. Now fructose 1,6 bisphosphate will now be split into two parts. One part we call it dihydroxyl acetone phosphate. The other one we call it glyceraldehyde 3-phosphate. Are you there with me? This one is called dihydroxyl. Let me just spell it. Dihydroxyl acetone phosphate. Are you there? Why this other one is called glyceraldehyde 3-phosphate. Now we've splitted it now. Now the idea is that majority of the time dihydroxyl acetone phosphate will be converted back to what? To glyceraldehyde 3-phosphate. Are you there with me? It will be converted back to what? Glyceraldehyde 3-phosphate. The only time whereby you will have more of dihydroxyl acetone phosphate is during starvation, whereby we want to form more of ketone bodies. But on a normal day, dihydroxyl acetone phosphate will form glyceraldehyde 3-phosphate. And before we proceed down the line, let me quickly tell you the name of the enzyme here. Remember, this is step 4. The name of the enzyme here is called aldolase. Aldolase. Are you there with me? It's called what? Aldolase. Alright. So let's proceed. Let's proceed. Let me take this a little bit down. You already know the name dihydroxyl acetone phosphate. So I will just remove this now. Like I said what? Dihydroxyl acetone phosphate will also form back with glyceraldehyde 3-phosphate, G3P. I'm using this to represent glyceraldehyde 3-phosphate. So I get my point. So both like this now we have two glyceraldehyde 3-phosphate. One here, the other one here. Are you there with me? Alright. This step 4 is what? The splitting phase of glycolysis. Splitting phase. Catalyzed by what? Aldolase. The next phase now from step 5 to the end, what we now call the payoff phase. What do we call it? The payoff. That's where you will now get your investment back. Payoff phase. It's called what? The payoff or phase. Payoff phase. Alright. So basically we'll go straight into it now. Or I will stop here now because this video is already 20 minutes. I will stop here now. When next we meet, we will continue from that payoff phase of glycolysis. In the payoff phase of glycolysis.